33A, or vadestetuximab talarine, is an antibody-drug conjugate. The antibody is directed against CD33. CD33 is an antigen on the cell surface of leukemic blasts in more than or about 90% of AML patients. It is linked to a powerful payload, a pyrrolobenzodiazepine dimer. That PBD dimer is able to bind to double-stranded DNA, cause DNA breakage and cellular apoptosis. The PDP dimer is not a substrate for multi-drug resistance efflux proteins and channels. The linker that holds that payload to the antibody is much more stable in the circulation than prior CD33 antibodies such as gemtuzumab ozogamicin. And then finally, the antibody has been engineered for more uniform loading of the payload pyrrolobenzodiazepine dimer onto the antibody. In a monotherapy study, a phase 1 monotherapy study, we found acceptable and expected on-target myelosuppression with the drug in patients with relapsed and refractory AML. We saw responses across the dose ranges, but the highest rates of blast clearance were seen with doses of 30 micrograms per kilogram given intravenously over a few minutes. And even MRD negative states were seen with that single dose of 33A.
Okay. And then of course we get to the remission rates and the toxicity profiles.
So given this promising information from the phase 1 study of single agent, we wanted to investigate whether the addition of 33A to standard AML induction chemotherapy of 3+7, 3 days of daunorubicin, 7 days of cytarabine, might add benefit and would be tolerable. So this was a phase 1b study where we did a dose escalation looking at the tolerability and safety of the addition of a single dose on day 1 and a single dose on day 4 of 33A to standard 3+7 in patients up to the age of 65 with previously untreated AML, including secondary AML in the study. What we found in terms of tolerability is we found expected and manageable, acceptable, on-target myelosuppression. The non-hematologic toxicity that we saw with this combination in the AML induction was similar to what we would have expected with 3+7 alone, and was generally just grade 1 and 2. We looked at the median time to platelet and neutrophil recovery and found that in patients who received 1 cycle of therapy and cleared their bone marrow at day 28, that the median time to a full platelet count or ANC recovery was about 4.5 to 5 weeks. In terms of efficacy, we found an overall response rate of 76%, 60% were complete remissions, and 16% of patients actually had a complete remission with incomplete platelet count recovery. Those responses were seen across karyotypic subgroups. What struck us the most about this efficacy data were the following 2 features. Number 1, that 30 of the 32 complete remissions were achieved with just 1 cycle of therapy. And historically, when we give 3+7, about a third of patients will require a second round of chemotherapy at day 15 based on persistent disease. So this was encouraging. It suggested more rapid action of the drug. And then the second encouraging finding from the study was that we did a flow cytometric assay for minimal residual disease in the responders. And what we found was that 78% of the patients were MRD negative using this flow cytometric assay when we said morphologically they were in complete remission. And that was including the majority, almost all of the patients who had a CR with incomplete platelet count recovery were also MRD negative. So in summary, what we found in this phase 1b study is that we could safely combine 33A with 3+7. The response rate seemed similar to what we would have expected with 3+7. In terms of the early mortality, it was only 2%, so it compares very favorably with 3+7 induction in this age group. But what was impressive was that the remissions were seen in 30 of 32 patients with only 1 cycle of the combination. And the second was that just over 3/4 of these complete remissions were MRD negative using a multicolor flow cytometric assay.